Next section. Here are some of the things that picornaviruses will do to the inside of the cell that will lead to more virus production. So this is one of the questions, and this is a nice thing to answer it. A little bundle of data here. So shown on this side, this little green guy is a nuclear pore, and it has lots of little proteins here, some of which you will probably remember from HIV entry because they were exploited, things like NUP 358, for example. Anyhow, these form the nuclear pore. It has a definite structure, and that's great. And when that structure is functional, you have transport both into the nucleus and out of the nucleus. So transport in is going to go via importin, alpha and beta. So anything with what's called a nuclear localization signal will get carried in by importin. And so that's going to be a thing that was used by HIV in order to bring proteins in to then bring genomes back out. There are also transportin 1 and transportin 3 pathways and the NRS-KNS pathway. So those are just all different ways into the nucleus. The main way out of the nucleus is the export pathway, which is going to involve CRIM1 and RAN-GTP, of course, is the thing that's going to help to power up that pathway. So, consider this from your average picornavirus's point of view. The cell is there to provide picornavirus with a home, a very short-term home, and lots of resources. And honestly, the virus does not care what happens to the cell after that. There's no co-evolution and live and let live. Viruses may not be sophisticated enough for that to really happen. So what these viruses do, so we've got two different viruses here, and so they have two mechanisms of doing essentially the same thing. So in addition to the stuff we talked about with translation, where EIF4G is cleaved into two little bits, we're also going to have the picornavirus 2A protease from poliovirus. And then rhinovirus has the 2A protease again, and also its 3C protease, which is its main protease, will cut some of these. And so you look for the appropriate colored Pac-Man, and you can see on pretty much all of these nuclear pore proteins, they are cleavage targets. So once the picornavirus protein, polyprotein, starts being translated, we're going to make some 2A, we're going to make some 3C, and then, at least for rhinovirus, the 3C and 2A are going to come in here and just wreck the place. They're going to cleave numerous nuclear pore proteins. So we got cleavage of NUP 358, NUP 214, NUP 62, and NUP 98. And then you get some of those same ones over here. Also cleavage of NUP 153. Those are going to be done by the 3C protease. It works a little differently when it's still attached to 3D. So it's 3C and 3D, like two domains together. That one seems to have a slightly different protease specificity, but the outcome is the same. By destroying the proteins that make the nuclear pore, the virus will actually, it would appear, collapse and clog all the nuclear pores very quickly. That means nothing can get into the nucleus and nothing can get out. How does the virus benefit? In a sense, the virus doesn't, because the virus is dependent on translation. Translation is dependent on a constant supply of ribosomal RNA from the nucleus out to the cytoplasm, because those ribosomes keep breaking down and you got to keep fixing them, or else, yeah, you run out of ribosomes. There are also other processes in the cell that require input from the nucleus. I mean, that's why the nucleus is there. What does the virus gain, though? Every response that the cell could make is going to involve some sort of signaling, which will then go into the nucleus, and then genes will be made, transcribed, expressed, first transcribed in the nucleus, spliced and sent out the nuclear pore. And then those genes will be translated and they can become things like interferon. They can activate various pathways, alert nearby cells, and essentially have antiviral effects. By destroying the effectiveness of the nucleus by making sure there's no way in and no way out, the virus prevents all of this signaling and all of this communication. The only things that are active are whatever is left in the cytoplasm. The virus depletes the cytoplasmic store of ribosomes and nucleotides and stuff like that. And at this point, the cell is dead to it because the cell is basically dead. And so the cell will eventually rupture. And the little viruses will come out. They assemble inside the cell and they just wait for the cell to die and burst open. Yeah, gross, huh? All right, the other side of this is that those proteases, the 2A and 3C proteases of both rhinovirus and picornavirus, like poliovirus, are also going to have effects in cleaving many cellular proteins that would be involved in making an immune response. There's no way you're going to remember all of these, and I don't know that it's to your benefit to remember all of these, but here, I'm going to show you. So look at all these targets for the 2A protein. When we get signaling by any of the interferons, so this is another cell that's infected, sending out a danger message and trying to alert this cell. That's great, but the signaling is going to go via JAK1 and TYK2, or JAK1 and JAK2. JAK1 is the common component there, and the 2A protease will actually cut JAK1. And so if there's no JAK1, there's no signaling from type 1, which is like interferon alpha and beta, type 2, which is like interferon gamma, or type 3 interferon, which are some of the weird minor ones. So that's a good way to prevent the cell from ever knowing that anyone else, is infected. It cuts the cell off from the world. Yeah, it is just a blockade. Then, if a little bit of that signal happens to get through, that's okay, because 2A is also going to cleave STAT1. And STAT1 is a common sort of a secondary messenger. It's going to get phosphorylated, it's going to go into the nucleus, it's going to bind to either a STAT1 or a STAT2. And again, STAT1 is the common component in all of these pathways. And so that's the component that rhinoviruses, the common cold, the world's mildest virus, are going to shut down. And they shut it down hard. So there's no effect of interferon coming from outside the cell. So you'd say, well, great. Oh, and the 3C protease will also cut TAC1 and IRF9, both of which are involved in some of this signaling. Just for thoroughness sake. Let's see. Over here, we've got some ways that the cell might detect that it's infected and might sound the alarm and produce a bit of interferon if it can before the nuclear pores get shut down. So, for example, MDA5 and RIG-I. The RIG-I is going to be cleaved by the 3C protease, and MDA5 is going to be cleaved by the 2A protease. So isn't that neat? MAVS is going to be inhibited by 2A, and we're not sure if that's a direct cleavage or if wrecking the MDA5 upstream is sufficient to shut down MAVS. So basically with these two proteases, they're very useful. They have roles in the viral replication cycle. 3C is absolutely required. And 2A has to make one cut, but that is a required cut, separating region 1 from region 2, if I remember correctly. And then 3C cleaves all the little pieces of region 2 and all the little pieces of region 3. So in region 2, we have the 2A protease. We have 2B, which does interesting things in the cell. And then we have 2C, which is a helicase-like protein. And then in region 3, we have the VPG, and that's either 3A or 3B. You'll have to look on the previous slide. And then we have the protease 3C and the main polymerase, which is 3D. So, very thorough. In addition to blowing up the nucleus, they're going to shut down very hard a lot of signaling mechanisms inside the cell. And remember, in addition to that, they are also cleaving part of the ribosome so that no cellular proteins can be translated by ordinary cap-mediated translation. It's diabolical, and it's kind of a miracle that rhinovirus does not just murder us completely. But yeah, our immune system is good at tracking rhinovirus down, and generally that doesn't happen. But it's a nasty little virus that is really messing with your cells. So there you go. Yeah. Thanks very much.